Okay, this video today talks to you if you are a marine ecologist and you have a lot of data, like data from reef surveys, which you would like to analyze in a more sophisticated way than you probably usually do this. So, what data am I talking about? You might have surveyed the coral cover of a reef or the fish populations or something like the macroinvertebrates, like the sea stars, or all of that together. Now, what would you normally do with this data? You would, for the lack of a better term, you would do a linear analysis. You would see, is there an increase, is there a decrease, is there some kind of trend, and maybe how do these variables correlate? Is it that if there's more coral cover, there are more fish? Is it if there are more macroinvertebrates, there are more fish or fewer fish? You would do types of analysis like this. Now, this is of course useful and this is insightful, but most likely there's more information in your data. And I would like to tell you about analysis methods which are summarized under time delay embedding or nonlinear time series analysis, which can get a lot of information out of your reef data, your survey data. Now, what do we do when we go about doing this time delay embedding data? We are making a three-dimensional object or four-dimensional object, five-dimensional object, or we're making a two-dimensional trajectory out of these time series of either your corals, your macroinvertebrates, or your fish. And then we're looking at the properties of these geometrical objects which we have created. How are we doing this? Number one. Okay, in the first example, we have a fish population varying over time in this oscillatory manner. How are we gonna make time delay embedding from that? So we will eliminate the time axis and instead we will plot the fish at time +1. So maybe that could be the next year or the next month against the fish at a point in time. And of course, then we need to have the same axis on the X and on the Y axis. And with that, we will just replot, and then in the first year we had 50, second year we had 100, so we just plot the different fish populations in different years against each other. We connect the points and then we have this trajectory now in 2D. So as a second example, we're using a wire. Again, the wire is a time series, and then we replace the time axis with the axis which shows the fish just simply the next year, and instead of having a linear wire, we have now bent this to a trajectory. And if we look at that in 3D, we see it's flat, so it's essentially a two-dimensional object. But that doesn't have to be the case. There could be additional dynamics. There could be a kink, and then we see that we have three-dimensional dynamics. Next example is the Lotka-Volterra model, which just models how a predator feeds on prey. And we can see both of them oscillate. The predator oscillates with a little bit of a delay. We can plot a time series of this, and then we can plot both variables against time going to the right, and we can just simply plot one variable, which is the predator, against the prey, and we get this trajectory in phase space. We have a time delay embedding. Now, why go through all of these complications with your perfectly fine time series data? The reason is that you can detect some regularities in these geometrical objects which you create from time delay embedding, and you can find out interesting things about the properties of your system. First of all, just the embedding process itself is insightful. The properties of the object which you create will change if you increase the embedding dimensions, if you go from two-dimensional to 3, 4, 5-dimensional. And at one point, these changes will plateau, they will stop. And this is probably something like the dimensionality of your system. So that in itself can be a very powerful insight. How many actual dimensions will there be governing your system. So obviously we talked about the different variables you were recording. So again, this could be 5 different types of corals you're recording, 20 species of fishes, 10 species of macroinvertebrates, but not all of these variables will act independently. It's very likely that if one fish species increases in abundance, another one will also increase in abundance. So, for instance, 2 types of plankton feeding chromis. Now, if you determine something like the dimensionality of your system, you will see how many really independent variables are there. Then you can determine things like, is this system which I'm actually looking here, which might look very noisy, is this actually noisy? Or is there a higher order rhythmicity? Is there what's often called a low dimensional chaos? So is this a deterministic system which just because of its chaotic nature looks very wiggly, it looks very noisy? Or is this actually a random process? Is this noise? Now, there are certain issues with this analysis. And these issues are that typically the bigger your system, the more complex your system, the more data you need. And for coral reefs, this is of course a critical issue because everybody knows, particularly you, if you're my target audience, that a coral reef is a very highly complex ecosystem with all kinds of species and abiological processes like temperature and carbonate concentration in the water interacting in a nonlinear manner. So the higher the number of important, meaningful variables in your system, the more data you need. And the type of data which is usually acquired in this service is, maybe once a year that you will do a reef survey. Or, if you're really lucky, every couple of months, let's say 4 times a year. So we are really talking about something like, in the best case, for instance, in the case of Apo Island, there are 40 data points over time from 40 years. This traditionally would have been not enough to do a meaningful time delay embedding or nonlinear time series analysis. However, in recent years, applied mathematicians have worked on this, and there are now tricks which can be used. And particularly, the tricks come down to if you have more than one variable, so you're not counting the abundance of one fish species, but of 30, you can twist and combine this, that despite having a relatively small number of time points, you can still conduct a meaningful nonlinear time series analysis. So please get in touch with me if you're interested in working on that. I'm actually burning to try these methods on some of the coral reef data, which my colleagues in the Philippines have acquired in recent years. The methods of nonlinear time series analysis are very transferable between different scientific disciplines. So I have published some work where I have used this kind of analysis for single cell neural data, so for spike trains emitted by pyramidal cells and interneurons in the cortex of rats and mice. And however, the type of mathematics and the type of approaches in these analyses are actually really similar, whether you're doing neuroscience or whether you're doing reefs. So I think there is probably a significant amount of data already existing, and what it takes is, us, you, the ecologist, and me, pooling our resources and getting additional insights out of this data. So I hope this was interesting. If you have any questions, please either contact me directly or leave a comment. I'm making a good effort to answer all the comments, and see you soon.